just I want to know about regulatory requirement. What is the limit for nitrite in my APIs or excipients? Like we have the nitrosamines limits are there, but what about nitrite levels in my excipients and APIs? Yeah.
There are no. Able to hear me? Yes. So there are no regulatory limit published for nitrate. So you see vendor to vendor, they are declaring different limits. Here. But some of the literature shows that if your nitrate limits are very lower, but actually some of the vendors are coming with nitrate-free excipients. Like if you have a 0.5 ppm nitrite, or if you have a 0.03 ppm, it will have a huge impact on the nitrosamine. So just to answer your question, there is no regulatory limit published for nitrate, as far as my knowledge is concerned. Maybe Dr.
So I can give another example here. This sodium azide. Sodium azide is manufactured by chemical reaction from sodium nitrate only. So this all the nitrosamine issue started with valsartan. NDMA coming from valsartan by reaction of this dimethylamine with sodium azide, which contains the sodium nitrate. So the process capability or the purgeability or the reaction condition is also critical to determine the nitrate level. So you can have high level of nitrate, but your reaction conditions are not favorable for the nitrosating reaction, then there is no issue.
Yeah, thank you very much. This is Dr. Tarak from the Ways. I have 2-3 questions. First one is we are manufacturers of contrast media. As a contrast media having high dose levels, the specs of nitrosamine or NDSRI is very less. With existing different vendor, high sensitive instrument, we are unable to quantify with that specification level with the 30% or 10% LOQ level. After we are putting so many efforts, there is any relaxation from regulatory bodies by providing these activities? What are the development efforts we are included during development?
So you are facing challenge in controlling the nitrosamine. That is the question, right?
No. As a API is a contrast media, and the dosage is very high. Based on the MDD level high, the specification is very less, PPB level. Existing high-end systems, Spinco, Shimaju and Thermo, all are having a high-end system. With that high-end system also, we are unable to develop method with that specification, to meet that specification level.
Okay, you are unable to detect the nitrosamine because of the high dose of the drug in the this available equipments.
Yeah, right.
That's correct, no?
Right.
So, I don't know which machine, because most of the machine available today, either from Shimatsu or from other manufacturer, can meet the current regulatory requirements. You can refer many publications of Dr. B.M. Rao also. I think you need to play around with the sample preparation, Like Dr. Manikandan has already explained, if the limits are very low, depending on the solubility of the compound, you can increase the concentration. Otherwise, you can do some precipitation or the, leaching. With sample preparation only, you need to play around if the machine cannot support. Sometimes you have this NDSRIs or nitrosamine where ionization will be very difficult. So that time also these kind of challenges will come.
Okay, second question to Manikandan. This is regarding sample dissolution. Because we are supplying so many customers API, our focus is compulsory sample should be dissolved in the preparation stage. To minimize that, we are using SP. Sometimes we are using sample spike standard for system suitability purpose. Is it acceptable? Most of in some HPLC methods with USP monographs it is published. There is any alternative rather than this?
So this sample spike method is already given in many of the monograph, but you need to, this sample dissolution is the best method. If you refer the examples given by Dr. Manikandan, so dissolution is must. Like if you determine a moisture content in any compound, if the sample is not dissolved completely or it is dissolved completely, you can imagine what kind of error it will, create for the undissolved material. So same kind of error can also come up in the nitrosamine determination. But you need to, experimentally find out the optimal dissolution required to achieve the desired accuracy level. And this spiking method, there are a lot of challenges with that spiking method in the sample. So when you prepare the standard, I came across this kind of method very recently, where surprisingly I found that in the sample and standard, same concentration is spiked. So preferably the standard you should use double the concentration what you spike in the sample. And you need to, critically evaluate, because if the concentration or the levels are very low, the area you get, how much error it can contribute. So that we need to do a lot of studies. So I think more to do with the method development and before you validate that method.
Okay. Thanks.
To answer your previous question regarding the sensitivity of the instrument, as Dr. Saji mentioned, one of the most important thing is if the intrinsic nature of the compound where the ionization potential is little large, sometimes you may not get that kind of sensitivity. Therefore, the conventional collision energies that you use, 35 electron volts, 40 electron volts, may not be sufficient. You have to go for a higher collision energy, even as high as 140 electron volts. You should be able to get the product ion, which will enhance the sensitivity analysis, where in one of the cases, we have applied 120 electron volts, where we have achieved sensitivity down to 6 ppt of an NDSRI.
So,
Keep the mic closer.
sir, in your risk assessment formula, sir, you are assessing the chemical thing, na, sir. In the flow chart, while you making methodology flow, na, sir, in that if you found something component is a very highly risk. So if there is any residual risk model or anything equation model can be substituted, or the risk can be reduced, sir.
See, when you do the risk assessment, nowadays everybody does 360-degree risk assessment.
Yes, sir.
Okay, you need to do a very, very comprehensive risk assessment. So when you do the risk assessment, first you have to identify the direct risks.
Yes.
Then there is an indirect risk.
Indirect risk.
So most of the time what we do is we do the risk assessment for the direct risk, and indirect risk, with our knowledge, we neglect, or, sometimes deliberately or unknowingly we do that. So sitting together all the CFT people with, chemistry team, the analytical team and the manufacturing team also, doing a brainstorming can definitely give a lot of insights that what are the real risks when, product is really manufactured in the manufacturing facility. So then it will give a more weightage, and then you can definitely reduce the risk. And there are a lot of NOx filters and amine filters are commercially available. Most of the pharma companies are using to reduce the NOx risk, which we can definitely remove to the not detectable level from the manufacturing environment. So if a risk is identified for the NOx level or the nitrate level, try to use these kind of filters commercially available to reduce the risk. If there is no direct risk, indirect risk you can mitigate using this kind of
So in the competency, MSDS is a material safety data sheet, like all kind of chemical compound if you mix it.
Only scientific justification, will not work. You need to do the confirmatory testing, because we have to make sure that the product manufactured is free of nitrosamine or the NDSRI, and we are, protecting the patient. This is the only objective. With this objective only, our scientific community should work. This is the only expectation from agency.
afternoon, all of you. I am Dr. Srinivasan from Orchid Pharma. Thank you all speakers, very nice, wonderful, insightful. My question is regarding NDSRI. In our API, when we do the risk assessment, we identify 5 NDSRIs. Out of that, 3 NDSRIs are stable. 2 NDSRI impurities are not stable because of the thermal or, during the rapid degradation reasons, it is unstable. In such a case, we are qualifying all the 3 NDSRI impurities and we are completing the analytical method development and validation. Now we go for the filing. During the filing, can we discuss this reason that during this risk assessment 5, out of that only 3 could be identified and qualified, and 3 is unstable. Giving the scientific report, can we explain whether it is regulatory perspective, this is acceptable?
See, I don't know whether you are carefully concentrated while Dr. Manikandan was giving the presentations. So he has given a nice case studies for these kind of issues. So if there is a possibility of 5 NDSRIs, definitely we have to demonstrate that we have a validated method which can accurately quantify these NDSRIs to the 10% AA level. And what you need to do is, I think the best way is that the 3 nitro NDSRIs, you develop one method and the other 2. Generally, if you look at nitrosamines are very stable. It doesn't degrade that easily. So you need to look at a method conditions which can keep that nitrosamine stable. So you wanted to add something?
Yeah, that's right. Actually, the first thing is before you conclude that it is unstable, you need to document or all your experiments, what you have done, to make sure that your experiments is actually true to your conclusion. Because before we say it is not stable, you need to generate sufficient data that can be analytical data or that can be a process data.
Process data only.
Yeah. So so if you're not able to even synthesize the impurity, I think there are some cases where if you document all the experiments saying that we are not able to prepare this impurity because of so and so reasons, and I think that experiment can be presented to the regulatory agency.
Yeah,
you can actually disclose this in your risk assessment report and conclude saying that because of this justification, but you need to properly document and summarize all your experiments, and you should be able to clearly say that this is not able to form. Because there have been some queries also that you conclude that it is not able to form, but somebody is already selling in the market. So that should not be the case. You should have make overall conclusive, very conclusive experimental compilation.
Fine.
Then that can be presented to the agency.
Yeah. Okay. Thank you.
Good afternoon.
All the impurities, as long as we have not synthesized, they are only theoretically possible. So if many times we will encounter that many of the NDSRIs are not possible to synthesize. So that experimental evidence with whatever MS scanning or the chromatograms or the NMR we have done.
Yes.
With that, if you submit to the agency will definitely accept.
Okay.
But once it is formed, once it is synthesized, then it is degrading. Then we need to identify the condition where these nitrosamines will remain stable during the during the testing.
Yes, doctor. My challenge is only in the synthetic part. It's,
Right.
Though we are theoretically arriving the potential impurity, we are unable to synthesis. That's the case I am talking about.
That will definitely, we can justify scientifically with the experimental evidence.
Yeah. Thank you. Thank you very much.
Hello everyone, and a great presentation by all the panel members. So my general question to all is, while we emphasize more on the storage and the synthesis based nitrosamine, however, what is our understanding on when a drug is given inside the body, let's say in vivo, because of the metabolic potential and the CYP induction, what kind of challenges it can lead to, and what kind of tool industry is using to upfront like look into such potential at in vitro level?
Your question is the activity of the NDSRIs in the body?
No, no, not activity. Let's say we have given then certain levels are there, and which is like panel members have very well covered in their presentation. But let's say after going inside the body, because of the SIP induction, if these levels are increasing, then how we should upfront look at the in vitro level itself so that later on it cannot be a challenge.
Okay. You are saying that once it's enter the body, there is a possibility of the NDSRI or the nitrosamine going up, which can increase the toxicity.
Yes,
So then we need to look for almost, ND level in the API or the drug product. And we also, I think this is the only way, and many times we can look for the, vacuum seal packing, which industry is using to prevent any air also coming in contact with the drug product and drug substance during the storage. So, anything you want to add?
Yeah, actually, Ravi, I think just to answer your question, if you see how these, the limits have arrived, right? The limit has arrived at a 70 years exposure. So it's a long time, right? If we really control the impurities well within the AA, right? What we have presented in the morning, right? For example, FDA 10% below AA. So I think the risk to the patient is very, very low, right? So basically all the guidances say below 10% AA, no need for any testing, okay? And in fact, for example, if a patient is taking a drug at the age of 30 years, I am not sure he is going to survive up to 100 years, 70 years, right? 30 plus 70. So if you see the LTL approach, less than lifetime approach, all these specifications are arrived based on the, 70 years of exposure, which is very, very high. So already they have taken into consideration, safety.
And I want to add one thing, I was just remembering while having a chat with Dr. Venkat. So the vegetables are rich source of nitrates. I think you all are aware. So when we take vegetable, we are introducing a lot of nitrates into the body. So if you take medicines, what you are referring, so there is a high level of, this nitrosweating reaction, happening, because in the stomach there is a HCl, it's full acidic only. So all conditions are favorable. So maybe, regulator may come up with having some controls on the vegetables in future. It is very likely, and the SpinCo team can also, proactively take up some testing for the vegetable also to the, maybe nitrites.
So Dr. Thomas, you are telling on lighter note, however, it is happening actually. And if you see ICH guideline, they have already mentioned, and as industry is generating understanding, that's why I wanted to bring into this discussion. So that while we are heavily emphasizing need during the process-based chemistry, synthesis, storage, and you rightly mentioned where the secondary and tertiary amines are there in the structure, and that under acidic stomach condition, there are the chances that such kind of things happen. And that is why you are seeing several cases because of this adulterated food and other aspects, so that's why they are increasing. Thank you. I stop here because I wanted to get into the discussion.
These are the areas, now agencies and the industry and other experts will start looking. Correct. These are the...
Good afternoon.
Yeah. Just only one comment. As our Dr. Brau and Dr. Saji mentioned, our body itself intrinsically generates NDMA at liver. And that is at such a controllable level, its genotoxicity does not affect the health as far as the carcinogenity is concerned. On the other hand, when they calculate the acceptable intake AA, as you rightly mentioned, the 70 years of age as a parameter to begin with, because those NDSRIs, what we know is, at present, we have not done any Ames test. Because when you do Ames test only, you can clearly say in 6 months' time whether it is allowed carcinogenity or not. Under a circumstances, by knowing the structure of the compound, as far as if the alpha carbon has been taken care of NDSRI, it is expected that this NDSRI is not supposed to cause carcinogenicity at this point in time. This is only an observation. It is not a criteria.
I have a couple of questions on NDSRI impurities. One of the NDSRI impurity reported in FDA guidance, that is dihydropyridine calcium channel blocker molecule. We are not able to synthesize the impurity in-house, and even we approached a lot of laboratories. So they are not able to synthesize the impurity based on the practical difficulty. So even it is mentioned in FDA guidance with a limit of 1500 nanogram per day. Can we still able to justify based on the efforts made in the synthesis of impurity?
See, like Dr. Manikandan has already explained, and we discussed also in the previous session, if you can scientifically justify the impurity formation is not possible based on the experiments you have conducted, then agency will accept. Of course, that impurity should not be available anywhere in the world. If some of the, there are a lot of companies, CDMO and CRDMO companies offering this nitrosamine impurities, NDSRI. So you should touch base with some of the companies and you get the well-characterized data before if you wanted to procure that. Otherwise, with experimental evidence, we can submit the documents to agency accept that. That is our experience.
Yeah. One more question. One of the NDSRI impurity in injectable product, it is crossing the limit of 1500 nanogram per day. Okay. It is due to the very trace level of nitrate present in the excipients. So, but none of the excipient vendor is able to control at a very low level of this nitrate. So we can't avoid this impurity. We performed the enhanced Ames test and proved that this is non-mutagenic, and we submitted with the higher limit, but still it is pending with the FDA. So is any hopes of approving the limit of more than 1500 nanogram per day based on LTL approach?
See, Dr. B.M. Rao, in the presentation, he has explained that accepting that approach from FDA is not very likely. But if you submit to the European agency, they may accept. And you need to seriously look for nitrate-free excipients, to make this injectable compound successful.
Yeah, but
Otherwise,
even reputed vendors like Merck, they are unable to control. They clearly tell that they are not unable to. They are not able to control nitrates at this level, at nanogram level.
there are a lot of companies offering now nitrate-free excipients. Yes, there are a lot of companies. So many companies are facing these kind of challenges, and they are managing currently with, in this interim higher level approach, and parallelly working on the technology to make it nitrosamine free products. So you can touch base with, the excipient manufacturers. They may be offering different grade,
Okay.
with a higher cost.
Okay. Thank you. Thank you very much.
Hi, good afternoon all. Thanks to panel, have depth insights on regulatory aspects as well as the NDSRI method development. So I have one question regarding this NDSRI method development as quoted by Dr. Manikandan sir. It's regarding choosing a diluent of acetonitrile. So it can mislead NDSRI content. I mean, the mechanism, any case study, specific case study regarding choosing the acetonitrile can mislead this NDSRI content. Can you have any of the case studies? Or,
if I understand your question rightly, like you want to understand whether we can use acetonitrile as a diluent?
Yes, yes. So one of the diluent you told that while choosing the acetonitrile itself, it can mislead this NDSRI content. Yeah. So do you have any of the case study kind of thing?
Yeah,
so,
we have one case study where we used acetonitrile in one of the drug product. It's not the API, it's the drug product. It's actually a oral dosage product, tablet capsule. So where we observed that when you use acetonitrile, we have seen that the nitrosamine is actually formed during the sample preparation. So we later on identify that because of some nitrate contamination from one of the excipient, and because of the combination of that nitrite and acetonitrile, we found that in that particular NDSRI, it is increasing. So we avoided acetonitrile, and we were able to get consistent results. See, what you need to study when you use acetonitrile is you need to make sure that the multiple preparations you are able to see consistent response. Whenever this kind of ubiquitous reactions is happening because of acetonitrile, consistency will not be there in the peak area response. So that is what I mentioned in my presentation also, that we have to actually study when you are using the different diluents. There is no restriction as such to use acetonitrile, but case to case it has to be studied, documented well, and understood properly.
Yes, sir. Thanks, sir. So here the residual nitrate can be quenched by the quencher as well as the antioxidant. No? So in that case it can avoid, right?
Sorry, come again?
So you are told this nitrate, no? So nitrate, residual nitrate from the excipients can be quenched using the quencher as well as the any of the antioxidants in diluent. So so that can be quenched. So the endogenous of an NDSRI can be estimated.
Yeah, you can take the approach. By whatever approach, what is important is you need to ensure that the diluent, with your diluent, you should get consistent results, and it should give accurate results. Because sometimes overestimation will lead to a wrong conclusion actually. So what we need to do is a proper study, case to case, step by step, it has to be studied. And yes, you can take that approach as well.
Yeah, thank you.
So the sample preparation is the most critical, activity for the LC mass analysis. So that we need to study thoroughly, identify the risk involved, and the, consistency, how we can achieve the consistency. And another very important thing, see, typically in quality control labs, few modes are not used in the GCMS or LC-mass sample preparation area. So try to have dedicated few modes for sample preparation and standard preparation. That will, take care of lot of cross-contamination and challenges associated with sample preparation.
Hi. Another one is a tea break. So we'll have a, no.
Okay, maybe we'll take the last question of yours.
Question. Yes. Good afternoon all. Myself, TSN Raju, and thanks for the panel for the wonderful discussion and the presentations. My question is slightly different. I mean, for the countries like US, Europe, Japan, Canada have their own regulations to ensure the safety of the people living in their countries. I mean, as an industry heads, I mean, what kind of reforms are the regulations our government, is planning to bring in India? I mean, to ensure the patients' safety and people living in India.
So, Raju, I think even if you see the Indian Pharmacopoeia, right? So in July 2022, they launched the new IP in Delhi. So if you see the general chapter 5.54, there is a chapter on the nitrosamines. So India also, there is a, guidance is there in the IP, and we should, honor the guidance and we should evaluate. So as a part of the ethical companies, for example, I worked in Dr. Reddy's. So as a part of the proactively, as soon as the IP published, they immediately checked all the metformins. At the time, metformin is a, it's a big drug, right? Tablet. So they, checked the level of NDMA in the metformin tablets proactively in the Indian market as well. So to answer your point, there is a guidance in the IP as well. We have to honor the IP and do for the products in India also, because it is not only because patients are same everywhere. We cannot differentiate that. We can take whatever the products we want, and we give very good products to the abroad.
I can see smile on Ravindran's face, because if it is implemented in IP, I think every 6 months they may conduct MS conference.
The last question, please. Yeah, the last.
Yeah. Sir.
Really the last.
Okay. Sir, my question to Dr. Rao. Sir, we are engaging in the advanced intermediate manufacturing. Sometimes we face something. Since our manufacturing process does not use amines or nitrite, but our API customers require the intermediate to be free from nitrosamine. Specific question like, what specific limits should be considered for nitrosamine testing in the intermediates?
That is based on the purgeability you can decide. See, three conditions are required: secondary amine, nitrate, and acidic pH. These are the three favorable conditions for nitrosamine formation. And if your intermediate is having a risk, and in the next step, based on the spike and purge data, you can decide what is the level you can have in the N-1 or the previous steps.
Yes, sir. Thank you.
Yeah. See, let me make it quick. Traditionally, API manufacturers give declarations for their nitrosamines. I mean, and if requested for the risk assessment, they generally say it's a confidential document we cannot share. So my question here is, when such declarations are given to the drug product manufacturers, are the drug product manufacturers also supposed to do the risk assessment, because that's also being done traditionally? Or are they supposed to do a complete analysis, doing a method development, as Manikandan mentioned, do a complete method development and an analysis of the samples, at least the process validation samples during the stability again, and then, depending on the results obtained, continue to do or not to do the analysis?
So to answer your point, if you see my presentation, the FDA is very much emphasizing on the vendor supplier qualification, right? So during the supplier qualification only, you need to qualify the supplier and make sure that, for example, your vendor quality assurance.
Correct.
Make sure that they will qualify the vendor appropriately, and you can gain the confidence on the vendors, right?
So,
because see, I cannot test all the materials which is coming from the vendors, right? I have to rely on the vendor, So you need to have a robust vendor qualification program.
So the question emanates there only, like most of the vendors are just doing away with the declaration and say that the risk assessment is a confidential document. So how about making it from a regulatory perspective that the risk assessment should also be shared to the vendor, shared to the customer?
No. As far as my knowledge is concerned, if you are buying an API from the reputed vendors like MSN or any big companies, right? Dr. Reddy's.
See, I'm not talking of the
full
big
risk
companies.
assessment report.
Yeah.
They are giving the full risk assessment report to the drug product manufacturer.
Yeah, okay.
Because as a drug product manufacturer, I need to have the risk assessment report.
Because this is more related to the NDSRIs, not the simple nitrosamine. It's more to the NDSRIs.
Today the risk assessment is comprehensive. It is both simple nitrosamine 7 and potential NDSRIs. So FDA guidance is very clear. Manufacturer should have the comprehensive risk assessment with respect to simple as well as the complex nitrosamines.
Thank you.
Thank you, sir.
No chance.